Welcome, everyone. We will just wait a second for Zoom to let everybody in. All right, welcome, everyone. My name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to this May 2024 webinar presented by ASCCT and the European Society for Toxicology in Vitro. I want to encourage you to visit our web pages to learn more about both of our organizations and how we can collaborate and how you can support our activities. The webinar is going to be recorded today, and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenter using the Q&A feature and the toolbar at the bottom of your screen. And we encourage you to put questions in there at any time during the presentation, and then we'll try to address them all at the end. You can also upvote and kind of thumbs up questions that you like, and that way we will take the most popular questions. We have also enabled closed captions. If you want to take advantage of that feature, you can turn it on or off in the toolbar as well. And hopefully you will say hello and let us know where you're joining from, and we will get started. Just a quick reminder of the ESTIV meeting, which is happening very soon in June. There is still registration available, so if you didn't get to do that yet, you can still attend. Please go to their website and check that out. And our fall meeting is going to be in October in Research Triangle Park, North Carolina. We are almost ready to release the program of oral sessions. We got some really nice session proposals and continuing education proposals, so we'll be doing that soon. And don't worry, you can still submit abstracts. We will be taking them for some oral opportunities and poster opportunities later in the spring. You can, in fact, you can submit your abstracts right now on the website. So next month, we don't have a webinar scheduled, but we have some in the works. So there's possible that one will come about. So stay tuned for that and join our mailing list, or you can check our upcoming webinars page for that information. And we also have scheduled a July 16th webinar to focus with 2 presentations from the BFR and USCPA on thyroid assay development. And so finally, today, we're going to be hearing about physiological maps. Our presenter is Dr. Luiz Ladeira. He is a postdoctoral researcher at the Biomechanics Research Unit, GIGA, Molecular and Computational Biology at the University of Liège in Belgium. He has a background in cell and structural biology, morphology, and physiology, and holds a doctoral degree in cell and structural biology from the Federal University of Viçosa in Brazil. He currently works on the Ontology project, developing physiological maps and mode of action ontologies to support the development of in vitro and in silico new approach methods to reduce the use of animals in chemical risk assessment. Okay, Dr. Ladeira, the floor is yours. I will stop sharing here.
Thank you, Christy. Let me do a presentation, and I guess you have it, right?
Yep, looks good.
Okay, thank you. Thank you, everyone, for joining. It's my pleasure to talk about my work here, about what we are doing in Ontology. And, well, we'll be talking about physiological maps and disease ontology maps, or you can call it mode of action ontologies, using a systems biology approach. So for the ones that are not familiar with Ontology, Ontology is called, it is an acronym for ontology-driven artificial intelligence-based repeated dose toxicity testing for chemical for next generation risk assessment. So it's a big name summarized in a very nice acronym on top that represents a big international consortium. We are now 19 partners spread all over Europe and in the US, with the goal of developing animal-free and human-relevant strategies for the prediction of chemical-induced toxicity. So chemical-induced diseases, conditions. And in OnTox, we are focusing on 3 main organs and 2 conditions for each one of these organs. So we are studying the liver conditions, kidney, and the developing brain chemical-induced conditions. And we rely as much as possible on available data, models, and methods. We are not conducting animal experimentation, but all the new methods that we are developing are in vitro and in silico methods that rely on this new data generated, but also on all available data that we can get our hands on. So in OnTox, we have 3 main frameworks to structure data. That is the physiological maps that covers the undisturbed biology, the QAOP networks, it's a quantitative AOP network. Also, we are also doing the qualitative ones to derive the quantitative from the qualitative. That includes the toxicology mechanisms, the toxicological mechanisms, but also the kinetic aspects of each one of the relationships between entities on this network. And also what we call ontology. And ontology here is not a semantic ontology for the computational biologists in the room, the bioinformaticians, but it's a graphical map that structure many different layers of data. I'll be showing you how we make this. And we will start with the physiological maps. Today we will talk about physiological maps and ontologies. We will not talk about the AOP networks, but in OnTox, the AOP networks are being designed and represented also in the same technical aspects as the physiological maps and ontologies. So let's start with the maps. So talking about maps, you are probably familiar with this type of map here. Here we have the city, the beautiful city of Liège in Belgium, it's the place where I live and work in the University of Liège. And we use this type of maps to get information about our environment, how things are structured, and to design best strategies to go from one place to another. So relationship between 2 points, the source point and the target, right? And we'll be building our map concept on top of this physical map concept. And for the ones with a biology background, you're probably familiar with this map here as well. This is a metabolism map that maps metabolism reactions. Of course, in biology, we don't have only metabolic reactions, but also we can design signaling pathways and gene regulatory pathways, networks that are not represented here in this map, but in physiological maps, we are trying to integrate everything in a way that follows the principles of this type of map here, in which you have a source and a target and the information of that relationship. So the type of entity that is catalyzing each one of these. So going further to the physiological maps, they are graphical representations when they are maps of these physiological processes or undisturbed biology. They represent these biological processes and their interactions and they put together different physiological knowledge from different sources in a standardized format, not only graphically, but also the data format behind the graphical representation. So the final product of these maps are machine readable files, not only the picture that you have here in a figure format, but also a format that can be readable by the computer, and the system can understand these interactions. They allow data integration and annotation, and they are built upon data integration and a lot of annotation here. And they are made to be human and machine readable. On the graphical aspect, we are using a graphical notation called systems biology graphical notation. There are different graphical notations to represent biological process out there, but we are relying on the systems biology graphical notation because it can provide you a type of network that is directed, sequential, and mechanistic if you're using the process description language. In Ontology, we are using these two languages. SBGN has these three languages in B, C, and D: the process description, activity flow, and entity relationship. And these are different from the classical interaction networks that you can build with any sort of omics data set, for example, with protein-protein interaction networks or gene-gene interaction networks, because it describes in detail the mechanisms of each one of the interactions. So you can get to know exactly what is happening in a certain interaction. And these maps have a specific architecture that I'll be showing you in more details in the next few slides. But we start these maps from a conceptual model. So it's a picture, a figure. You have here an hepatocyte with a liver in the top right side. This is the conceptual model of one of our maps, the liver lipid metabolism map. And it acts as a sort of a small map, a mini map to the big map. In the interactive platform that we use to visualize and explore these maps, this conceptual model is also interactive. You can click in the name of a process there in the conceptual model and be redirected to a place on the main map that describes the molecular interaction in more details. We have the main map that uses the SBGN notation, right? The SBGN language to describe it graphically. And we have submaps that describes in more details what's happening in each one of the pathways that are represented in the main map. As we have three different organs and different maps covering different processes there, and different teams of domain experts helping with the curation of the information that goes on each one of these maps, you can imagine that we have different decisions, different curation decisions for each one of these maps, and they are slightly different between them. We'll be talking about each one of them and some specificities of these maps. So we have these three organs, but we have 5 maps. So we are studying 6 conditions, but 5 maps because of the size of these maps, and because also these maps need to be human readable, so the user can profit from what is represented there very easily. They cannot be big like network can get omics data set, for example. So as we are studying the liver steatosis, chemical induced steatosis, we have a correspondent map that is the liver lipid metabolism physiological map. We have a case study in Ontology for the liver cholestasis. We have a map for the biosecretion process. For the kidneys, we are studying kidney crystallopathy and kidney tubular necrosis, but we are covering these two conditions with the nephrine physiology map. And for the brain, we have two different conditions. One is from the very first weeks of conception. The brain maps are developmental maps, so they're slightly different from the other ones because they have time as a big aspect to be considered on these maps as well. So we have the neural tube closure map covering the first 2 to 4 weeks of development, and the cognitive function development, that is a brain development map covering from the week 4 until birth. I'll be talking now about each one of these maps and give you an overview of what you can find on these maps. So the first ones are the liver maps, and here you have an overview on the liver lipid metabolism physiological. I already showed you the overview, that is an hepatocyte with the different processes that we are representing in the map. This map has this architecture structure that you see in the top right corner. That you have a top level overview, a main map using BGN process description, and submaps also using SBGN process description. And this map is being developed in collaboration with the Liège University in Brussels, with the team of our coordinator, Mathieu Winken, and it contains all the important pathways to understand the development of this steatosis in hepatocytes. So we have cholesterol biosynthesis, the cholesterol biosynthesis pathway, fatty acids and cholesterol transporters in hepatocytes, glucose metabolism, fatty acid omega oxidation, beta oxidation in peroxisome and in the mitochondria, and so on. But we have also control pathways like a gene regulatory network, but also hormonal control of these processes. Until now, we are covering glucagon and insulin, but these maps, as you'll see in the next slides, are living documents. So they keep growing as we get more information. So every time we have a new piece of information that can be put together here, we update the map and keep this cycle going forever. And we have our second map is also being developed in collaboration with the VUB in Brussels, but with a different team of experts. It has the same architecture as the previous map, but it covers different mechanisms because this is for bile secretion physiological map. The first map that I showed is a single cell map. So we are covering only hepatocytes, and everything that is represented here rated for hepatocytes. So you can go to a pathway database and get the cholesterol biosynthesis pathway, for example. But that would be a generic pathway in most of the cases. What we are doing here is taking these pathways from these databases and from the description papers and curating for specific organ contexts. Because this map is mapping only the processes in hepatocytes, it's a single cell map. But here in the biosecretion map, we have two cell types. We have hepatocytes and cholangiocytes. So everything needs to be curated for these two cell types. So if we are representing the same pathway in these two different cell types, it needs to be checked to make sure that all the proteins that we are representing on the pathway are expressed in each one of these cell types. Here, as it's a biosecretion physiological map, it covers all the processes related to bile acids production, secretion, and reabsorption and recycling as well. Brain map, the brain map is a developmental map, so this is slightly different, and also the way we are organizing this map. We have this overview that covers the type of cells. We have many different cell types here to cover, and also different processes that these cell types are going through. And this map is organized as a top-level overview that works the same as an interactive mini-map for the big map. But in the main map layer, we have a cell-cell interaction map using activity flow. So if you remember from the SBGN slide, activity flow is not mechanistic, or at least it's not mechanistic enough from the systems biology perspective. But it's good enough to get us an understanding on how these cells communicate with each other and influence these processes on each other. And then in the submap part, we represent in much more detail each one of these developmental processes for each one of these cell lines. So the submaps represent each one of the cell lines that we are including on this map. The neural-tooth closure map is also a developmental map, so it also covers different developmental time points. As you can see in the overview picture here, each one of these blocks covers a different time point in the development and the closure of the neural tooth. And this map is mainly developed by my colleague Alessio Gamba here from the University of Liège, and the team working from the University of Utrecht. We have Job as our main domain expert. And for the ones that follows this webinar series, you probably have the opportunity to watch Job talking about this map and the work he's doing in modeling this map as a dynamic model last year, if I'm not wrong, in November. You have here at the bottom of the slide a link for the previous webinar, where you can get more details about this work. But this map is also designed following the top-level overview, process description architecture. Each one of the submaps contain a detailed representation of the most important signaling pathways and metabolic pathways for this neural-tooth closure process. This is a specific case of map that is already published in Ontology, so I can show you how the map looks like in full. You have here on this slide the published version. We are working now on a new release of this map, expanded release. And this was done by Job's advisor, Harm Hilsenfield, also from the University of Liège. And here on this slide, you also have a link for the interactive platform, the Minerva platform. So you can interact with the map and get the machine readable version, annotations, and everything. And finally, the kidney map is Metron physiological map. It's also being developed by my colleague Alessio, also in collaboration with a different contact team from the Utrecht University. As we have Devon, PhD student from Utrecht, as our main domain expert here. This map is also being built as a top-level overview. I will not show here the top-level overview, but also process description and process description in the submaps. And you have here on the left side a table of contents for each one of these submaps and the pathways that are present in the main map here. And you can also get more details about this map on the previous webinar from last year, because Devon also shared the webinar, so you can get more details of this work on the link below. So how these maps are built, I talked a lot on what are the contents of the map, but let's see how we construct these maps. as I told you, these maps are being built in close collaboration with domain experts. So I showed you the main contact people for each one of the maps, but we have a much bigger team of domain experts helping the curation of the information for each one of these maps. And this is done also with the support of large documentation and guidelines that we wrote together with the domain experts to put everyone on the same page and construct a more comprehensive tool that can be used by everyone. Our workflow is deeply inspired in the disease maps project. So it's a big community that constructing this sort of map for different conditions. They are mostly used for therapy exploration. I would say that the most famous map is the COVID-19 map that they did during the lockdown. And yeah, it's really worth it to take your time and explore their website. I have a link on one of my last slides. But this workflow that you see here, it's a loop. As I told you, every time we get more information on these physiological processes, this information can be included on the map. So we have a loop process that never ends. We start with this documentation set and documentation framework, the maps and our ontologies as well, that includes these guidelines and quality control checklists, a glossary, because on these curation teams, we have people from many different backgrounds, wet lab personnel, dry lab personnel. So not every time we are talking the same language. So we start a glossary to help everyone understand each other. Planning documents that work as a mini project for each one of these maps, and a table of contents that was agreed before we started mapping, actually curating and mapping each one of these processes. And from the planning documents and the table of contents that guides how the maps are curated and constructed, we start the curation phase and we have the physiological map. These physiological maps are being built based on literature. So we have a literature repository that is a Zotero group that we include all the references that we use. The maps are there, organized and annotated also. And then after completion, these maps are stored on the Minerva platform, that is an interactive platform. I'll show you in the next slides. On GitHub, mostly for the unstable releases, unstable version, the working version of these maps, but also on biostudies for the final stable releases of these maps. So we can have a stable identifier attached to these maps and make them more FAIR principles compliant. And after release of these maps, we have reports on the documentation as well. So we have release note report that describes what was done for this version of the map, what changed compared to the previous version, for example, it's like in software release manner. We have an issue report that is basically done by the curators, by our team in Liège. When we don't understand how a certain thing should be represented, or when we need help from the domain experts, we describe it there. And then after a while, we send these issue reports to the experts to get more clarification on how to represent a certain process. And we have these pathway discussions as well. That is where we register curation decisions for the specific pathways. These maps are built also in a modular structure, as I showed you. So this modular structure allows easily the update of the map. So when we get more information, we don't need to update the whole map, but we can update the submap, for example. So it simplifies things for the curation team. It improves the data visualization as well. It makes easier to understand what's happening on a system when you modularize things like we're doing. And it simplifies the review and the curation process because these maps are constantly being reviewed by the domain experts as well. And the modularization also allows us to push these into wiki pathways, for example, to democratize what we are doing, to make the pathway accessible and reusable by our teams around the world. The annotations that we are doing are being done manually by the curation team, by us in Liège, while building the maps, but we also rely on automated annotation as much as possible done by the platform. This is linked to the guidelines that we have set in the beginning of this mapping process. And of course, the documentation and the storage that I already mentioned. So the first step after setting the scene with the domain experts is deriving a purpose and scope for each one of the maps. So here we represent this, we register this on the planning document and in the table of content. So the planning documents contains the description of who will be working on each part of the map, who are the domain experts, who are the curators, why we are building this map, for which purpose, and this helps putting limits to the level of detail and to the mechanisms that we need to represent there. What will be on the maps, map content, relationship pathways, and key molecules that we need to include, but also describing the first references. The first version of these maps were built based on review papers and book chapters selected by domain experts. Now we are hopefully moving to the systematic review phase, where we'll be including more experimental references, experimental papers on these maps based on a systematic review approach. And the planning documents also describe how the map will be constructed, the level of granularity, and how we will sustain the development of this map. Here on the right side of the slide, you have an example of a table of content. So we are not only relying on literature, but also on what was already built before. So we are recurating and reusing pathways from pathway databases like Reactome, Wiki Pathways, KEGG, and our disease map modules, for example, of course, adapting and curating for a specific context of our maps. For example, this table of contents you see here is for one of the liver maps, and I'm using one of the COVID-19 modules, submaps that they have there. It's the mitochondrial metabolism. Of course, this submap was checked and reviewed for liver specific scenario and removed all the disease condition related relationships there. The next step would be data collection and processing. Well, this is the step in which we select the papers, the books, the references, and we screen each one of these databases and disease map projects to try to find blocks that we can reuse in our maps, models that we can use in our maps. In a second step, we'll be using a systematic review approach assisted by AI. We'll be using the SwissRev system to accelerate the screening of literature and also to translate the information from the literature into graphical mechanistic relationships that can be that can enrich our maps. And in the curation phase, we also define graphical representation. The graphical standard we will be using, it was when we decided to use the SPGN process description and activity flow, depending on the level of information we have to describe each one of the reactions. So if we have enough, we go for the most granular possible, that is process description. If we don't have enough information to describe in such details, we go for activity flow. And also the tools and the infrastructure that we'll be using to construct these maps. So we are using a software called Cell Designer to design these maps that can be SPGN compatible, not 100% because Cell Designer uses a very previous version of SPGN, but with some trick we can get them these representations full compliant with SPGN. Data format is SBML, but it's a Cell Designer specific format of SBML, so it's not a traditional SBML, but these are convertible formats that using the Minerva platform we can convert to different systems biology formats. We curate the literature, so we go paper by paper. As we started from review papers, we get a list of these review papers, and then the curators, our team here in Liège, go paper by paper, extracting mechanistic information. As you see on the top left side here, we identify pieces of information that will be put together and translated into a graphical representation. And then you have a small piece, a reaction that can be assembled in a bigger map. So it's like assembling a puzzle. We construct each one of the pieces, and then we put the pieces together in a bigger context. This is the big physiological map. This is how we take from the literature, but of course we are also relying on already curated pathways, as I showed you. The next step after the graphical representation and the map building is the map visualization and the sharing and review phases. And for that, we are relying on the Minerva platform, that is the molecular interaction networks visualization platform. It's not only visualization, it's also exploration, as I'll show you in the next slides. It's a user-friendly interface. It looks like Google Maps. It works like Google Maps as well. And in fact, you can choose to use the Google Maps API if you want in the Minerva platform. So you can get the same structure, the same sort of map of navigation that the same experience that you get from Google Maps. You can do it here with these physiological maps. The platform allows also for content exploration, so you can search for entities and information on the map very quickly, but you can also search for information in other ontologies, formal ontologies, like drag bank, and the CTD for toxicity information. And the Minerva platform also allows for automated annotation if we are using a standardized name for the entities. And last but not least, the conversion of formats. So we upload our maps as a Cell Designer SBML, but Minerva can convert into the standard SBML, but also GPML that's used in unique pathways, SBG and ML that can be used in different editors. But also if you go from one of these formats, you can go back to Cell Designer SBML. So in Minerva platform, this is an example of a map in the Minerva platform. You have different layers here. Let me take a laser here. You have this on the back is how you see a map first when you first arrive on the Minerva platform. So as you zoom in, you'll have this representation. These boxes start fading and you have a more detailed representation of a pathway. And when you click on a node, for example, you have the annotations for that specific node. And when you click on a reaction, you have the specific annotations for the reaction. Most of the times the reactions are annotated with a persistent identifier, or it could be a DOI or a PubMed ID, or the source, the paper, the piece of literature that describes that interaction. And after uploading based on the Minerva platform, you can send, Minerva is a web service, web server. So you can get a link for your map and send to your collaborators for reviewing. So Minerva also allows commenting, so you can include comments on the map, on entities, on reactions, and where. And these comments help us to correct the map, to update the map, and to keep this iterative process of improving and making them more comprehensive. So this is the first layer of information we have to construct what we call an ontology map, or mode of action ontology that I'll be showing you now. So for each one of the maps that we have, the physiological undisturbed physiology map, you have in orange here, we have a disturbed physiology, or a chemical induced condition matching each one of these maps. So in Ontology, we are studying steatosis, cholestasis in the liver, blood necrosis, and crystallopathy. I'm sorry for the mistake here. Crystallopathy in the kidneys, and neurotube closure defect and cognitive function. So the ontology maps are 6, one for each one of these conditions. And they work more or less the same as a physiological map, but here now in the ontologies, we can use these maps also to visualize on-demand data. You can overlay data sets and see which pathway are more active or not, which pathway are more busy. You can understand targets from your network and why these targets are leading to a certain condition, a certain phenotype downstream on your network. So you can zoom in and zoom out and get more detailed information, the same as in a Google Maps. But you can also click on a certain entity the same way as you do on Google Maps and get annotations and other layers of data from that entity. So I'll be showing you now on the Minerva platform and how it looks like. So here you have a view of a very first version of the steatosis ontology. So it's based on the liver lipid metabolism physiological map. And when you get there, you have this main layer of physiological map, but you also can visualize the overview model, that conceptual model, if you click here and navigate this big map through a minimap that is hidden here. You have this overlay tab, submap tab, and info tab to check, to be able to overlay your own data sets from your experiments on top of the map. You can check the submaps from this tab, and there is metadata related to this map or this ontology. And I have the search tabs here to search for drug interactions, chemical interactions, and microRNA interactions. And if you want to visualize the comments that anyone can include on your map, you just allow it checking these comments. on this side here. So zooming in on the map, you'll get this on-demand data. If you click on a reaction, you have the annotated data related to that reaction. So here it's a paper describing this specific reaction. If you click on a node, you have a linkage to the main systems biology ontologies. So it will be linked to gene-related ontologies, protein-related ontologies, chemical-related ontologies, for each one of these specific nodes. If you search for a drug or a chemical using the search systems here, you tag all the known targets for that drug or chemical in this way, and you have this small tag on top of the target here, and if you click there, you'll have on the left side here a lot of annotation on a lot of information on this interaction. If you scroll down this page, you'll have also the references describing the interaction of a certain chemical that you search it for with a specific target. So this is really helpful for talks to understand the relationship between chemicals and the targets and how they drive modifications in the activity of these pathways. You can overlay your experiments, I told you before. Here we are visualizing an overlay of proteomics data set for expected more healthy, I would say, RNA levels in hepatocytes. Single cell RNA data set from the Human Protein Atlas, but you can also visualize differentially expressed genes, protein levels, anything that, any type of information that you can match with a color gradient, you can visualize on top of your map. You can also map our source of information in which you have annotated a target. So here, what we have developed together with the University of Luxembourg team, that is a team that developed the Minerva platform, a plugin for the platform. It's an add-on that can read tables and map the information on these tables on the map itself in the same fashion as you see here on this slide. So here it's not our plugin, but our plugin works exactly the same. Here is a plugin called Drug Reaction plugin. It's inherited from the disease map community. And what you can see here is a table of information containing the name of the drug, the sort of information you have, if it's an adverse reaction or a warning, all the adverse reactions that you can get from this drug, and the molecular targets for this drug. That is the link from this data set with the map itself. So you can map there this sort of table, tabular information that you can annotate with a target. And we are doing this in order to annotate different induced conditions, for example, for the brain development map, because it's a big list of cognitive function and conditions. And a last feature that you can get from the Minerva platform is also visualizing 3D structure of the targets that you have there on the map and on the ontologies. This is a built-in function, so it comes by standard with the Minerva platform, and you can query and visualize the 3D structure of different proteins in the map just using their HNC standardized name. So to search for the matching UniProt ID, and you can visualize different structural information here for this protein, but you can also download this structural representation and use this for our modeling purposes, for example. So these ontology maps, as we call them, are these multi-layered tools as we have on top of the biological layer, that's the physiological map, many different layers of our information that comes from the different teams in Ontology. So we serve as a framework for data integration. And together with the adverse outcome pathways, the AOP, that are also being represented using SBGN in OnTalk, so they can be included as a submap in each one of these ontology maps. They allow this data integration among all the domains of knowledge that we have in OnTalk: chemical, kinetic, toxicological, and biological layers, using all this infrastructure that I showed you. And to conclude, well, we have these physiological maps. We start from the physiological maps to map as much as possible the undisturbed physiology, and then to try to understand how a chemical can perturb this physiology. So these maps are these graphical representations of biological processes. They can be reused for many different purposes, not only for toxicity assessment. They rely on standardized notation that allow for human and machine readability and interpretation. And they also rely on a lot of documentation and guidelines for the curation and annotation, making them more principles compliant, so we can make them more democratic. And the ontology maps are founded on these physiological maps, but also including the toxicological layer represented by the AOPs, chemical information, kinetic, and convex data as well. And in OnTalk, they will also be used for mechanistic risk assessment. And as you may notice it already, this cannot be done by one single team or just a few teams together, but it needs a large community effort. That's also why we are going to make these maps and pathways available on each pathway, so we can accelerate the curation of these pathways and make them more comprehensive, and they can be reused by everyone. I mentioned in one of the first slides here the disease maps project. We are also part of the disease maps community now, so our maps have a dedicated page on the disease maps website in which you can get information, and also the navigation link for the neuroTube closure, for example, that you can follow with any update that we have on these maps. We are preparing the publication for the other 4 maps, so that we can start sharing these maps openly with everyone. And we have recently prepared some tutorials on how to get started on physiology mapping that you have, and also how to use the cell design platform to make it easier for our teams also to start building these maps and helping curating ways that can be reused for different purposes. You have a link on the bottom here for the OnTalk CV, but you'll also find more tutorials on modeling, webinars, and meetings, open meetings that we have on the project, so you can get up-to-date information on how OnTalk is moving to achieve this mechanistic risk assessment based on artificial intelligence. And well, I'd like to thank my team, that is the curation team of this map, our supervisor, Professor Luiz from here, University of Liège, our manager and colleague, Bernard Stomon, and Alex Gamba, but also all the domain experts that you see listed here, and the University of Luxembourg team, that is an external partner, and they help a lot in developing more features for the Minerva platform that can serve our purposes, but also discussing relation aspects of these maps with us. And of course, the full Ontology team, because this needs a village to create these maps. And if you want to talk more about these maps and ontologies, of course, you can contact me at my email. And thank you for your attention. I'll be happy to answer questions.
Thank you very much, Luis. We do have quite a few questions coming in, so we'll try to get through them relatively quickly. So there's two questions asking about quantitative information. The one question is, do the physiological maps contain quantitative information about their representation links? And also, another question is wondering, how would you go about doing a dose response analysis using these maps?
Okay.
Any ideas about that?
So first, quantitative information. The quantitative information here comes from the omics datasets. So if you have an omics dataset that describe in a quantitative fashion protein levels, for example, or RNA levels, you can match these with the nodes on the map and have the quantitative information for a specific condition in which that data was generated. So that's why I showed you here the layer of omics with the human protein atlas for undisturbed liver RNA levels, because for the physiological maps, we use like control data sets, the undisturbed data sets. So if you want to visualize a quantitative aspect of a chemical induced condition, you go for a data set after disturbance assay, for example. And the second question was on modeling, right?
Could you use the information to do a dose response analysis?
Not directly. These maps can be used to derive models, dynamic models. So as you go for PBPK models, you have here the description of the physiology that you can use to support your modeling approach. And then you can go for those response assessments. Something that we are starting to develop here is the modeling of the whole network directly. From the disease map community, we have some tools that allows us to convert these maps into logical models, so Boolean networks, and that you can simulate different perturbation aspects, but still not a dose response modeling. But it's something that we are exploring how to get there.
Right, makes sense. There's a question about, well, they're making the comment that the epistemic knowledge these physiological maps represent is very important to model development, of course. And they're wondering, and you mentioned, of course, sort of standards and principles for their development. I guess it sounds like the team has developed those as part of the process of developing the maps, but I'm wondering if they're made publicly available, or they're intended to be made publicly available at some point.
You mean the documentation of these maps
Yeah.
and guidelines, right? Yes, they will be publicly available. We are just finalizing a perspective paper on this physiological mapping framework, and all the documentation and guidelines will go public as a supplementary material.
Great. There is a question about whether there's an like a website or an online resource available that can guide someone through the process of installing the software and the data sets so that they can use these physiological maps? Or how does that work if they wanted to?
There are some guides from the disease map community. So if you go, I guess I'm still sharing, right?
Yep.
So if you go to the disease map website here, you have on the bottom of the slide here, yep. You'll have some information on how to start a new map. The process is almost the same. The difference between our framework and the disease map framework is that the disease map focus on disease conditions, so disturbed conditions for the map. Right. And we are mapping undisturbed physiology. But the workflow is almost the same. So you can follow the steps from there, get the guides they had published recently on how to start a map, how to construct these documentations, the planning, how to interact with the main experts, and so on. And well, of course, you can get in contact with the leaders of the disease map projects, but also with us if you want to start a new map, and we can put everyone in contact in the loop, and you can be part of the community as well, attend the periodic meetings, get in contact with people that already done this before, get help. Sometimes it's not easy to learn from the papers itself. Sometimes it's not as clear as talking with someone that did this before is, right? So yeah, it's a way to learn and get to start in business.
Great, thank you. Maybe we'll take a couple last quick questions here. Can you say more about the process of integrating with AOPs? How does that work?
Yes, now we are working on a different representation of AOP and AOP networks. Traditionally, AOPs are represented as figures, right? Mm-hmm. You can construct an AOP using any type of graphical editor. And they are not traditionally machine readable, so we cannot really integrate these with what we are doing here. But there are some efforts starting on constructing AOPs for a while now, mostly from the Wiki Pathways community, using the GPML standard. But as we are not using GPML in Ontology, we started designing also these AOP and AOP networks using SBGN. So we are preparing also case studies to showcase how it can be useful and how you can take advantage of all these infrastructure that I showed you, also to explore AOPs and AOP networks and how to derive a model from an AOP and an AOP network. So yeah, it's like constructing a physiological map, but with the AOP information.
Great, thank you. Okay. Sorry, a few questions keep coming in, so people are still interested. If it's okay, keep going.
Yes.
All right. So one person is asking if you know of, or are there agencies in the United States working on developing similar types of maps, maybe of different biology?
If there are agencies in the US.
Or just maybe groups, I guess, anybody who might be developing similar physiological maps.
Well, you'll find these maps on the disease map community mostly, but if you search for SBGN representations or machine readable and type sort of machine readable pathway representation, you'll find a lot of resources. Also, Reactome is one of the biggest and maintained by the European Biomechanics Institute, one of the biggest pathway repositories. but Reactome has a curation gate. I would say it has a review process, internal review process, very tight review process, differently from Wikipathways. Wikipathways works as Wikipedia, for example. You can create your profile and you can include your pathway there, and you can help curating other pathways as well. So you can get these pathways from Wikipathways, you can get from Reactome, you can get from KEG, you can get from Pamphlet. There are a lot of pathway resources that you can find pieces of maps, but for this scale of map, you need to start putting the pieces together and build on the context that you want to study.
And I'm pretty sure you mentioned this, but I will kind of just make sure. They're asking whether there's a common platform where all of these physiological maps will live, and is that available now, or is it going to be available soon?
Yes, that is the Minerva platform.
Yeah.
I guess I have the link here somewhere as well.
Oh, you missed it.
Here, no, it's here. You have this ontalks.elixir/luxembourg.org/Minerva. If you go there, you'll not find the maps now because they're not available yet. We're waiting for the publications that will come very soon. But it's the Minerva platform that is the main platform that we share these maps. And our disease maps from our projects are also being shared through the Minerva platform. So if you go to the disease map community page, you'll find links to many other different maps from there.
Great. And just so everyone knows, these slides will be available on the web too, so you'll be able to click the links once we put them up. Okay, there's a question about the curation phase, the literature creation. Do you check the quality of the papers, the data resources, and how do you do that?
We have a quality control checklist, but this until now, this quality control checklist is mainly focused on annotation aspects, not the risk of bias from the papers, for example. That's something that we'll be including in the next phase because we are now basing the map curation on review papers and book chapters. So this is not as easy to check risk of bias from this sort of papers. But when we go to the experimental papers, yes, we'll be following something that can be implemented, risk of bias for these papers. Yes, but something, a reaction, a representation of a reaction is I guess it doesn't follow the same risk of bias as conclusion of an experimental paper as well. Because if you have this reaction being described from many different sources using many different methods, you can be sure that this reaction is happening and you can represent it with a certain level of confidence.
Yeah, that's an interesting point to keep in mind. This is maybe more of a comment than a question, but they're asking about knowledge that commercial organizations hold that won't be shared. I don't know if you've had any experiences like that.
I don't have experience with private companies, but I know that they have much more information than we have available openly about this mostly experimental data. But yes, this is information that we cannot include in these maps.
Yeah, I know the AOP community has the same issue as well.
Yes, yes.
Okay.
Unless you're doing this sort of mapping for a private company and you can get hands on the data that they have to support your map. But I would say that the problem is that you'll not be able to share your map openly, right? They will use your map for their interests.
There are some companies that have made systems biology maps and AOPs and published them, but not as much as data is available, of course. And then the very last one, thanks for your patience, is what funding options are available for large-scale community efforts needed? Now, obviously, you're working in a funded project.
Yes, well, I can tell from Ontology and from what I saw from the other disease map project. Ontology is a Horizon funded project, so it's a lot of it's a big budget, but this budget is not fully on the mapping. So we have a lot of wet lab personnel as well that needs much more money to buy lab supplies than we do to map this map. But these projects are not only for mapping. In all the cases, they have a different goal. The map comes as a supportive tool for math goal. So it's to support with physiology information, with mechanistic information in our case, to design the in silico models, but also to help find a better target for an in vitro assay, a new vitro assay. And in the disease map community, it's mostly to try to find targets for therapies that they can model as well, and so on. So it's always, it works as a first step to derive more solid hypotheses or scientific question that we want to solve.
Yeah. It seems like it's funding with a purpose. So it's developing the maps in order to help develop more models. Well, that's all the questions we have. I want to thank you very much for your presentation. It was very interesting. There's a lot of useful work that I think people will find impactful in a lot of different activities on, down the applications, different stages. So thanks again. Thank you all for listening, and hope you have a great rest of your week.
Thank you.
Thank you. Bye-bye.